Hello. Today we're going to talk about factors that influence hemoglobin saturation. So we're currently looking at an oxygen hemoglobin dissociation curve that shows us how hemoglobin saturation changes according to the amount of available oxygen. And our dark line in the middle is our normal curve. But as you can see, we have a dotted line above and below it, and that shows how the curve can shift based on various factors. So let's talk about what can cause each shift. So a shift down here to the lower line, what would that mean in terms of oxygen content? Like if normal was right here and then it shifted down to here, is hemoglobin going to be bound to more or less oxygen? I hope that you're saying less. So if we shift here to the right, there is a reduced affinity. Oops. Reduced affinity between hemoglobin and oxygen. And even though it looks kind of down, this is considered a right shift. And then let's think about conditions that could cause this. So when would we want to unload oxygen? That would be tissues that need oxygen, perhaps because you've been working out or something, so your muscles are low on oxygen. So that's going to be one of our conditions. And then what else do you know about tissues that have been working really hard? Like if they're low in oxygen, they will be high in CO2 because that is a waste product of cellular respiration. And then a couple other factors that go along with the high CO2. How does CO2 leave the body? Some of it's dissolved in plasma, some of it binds to hemoglobin, and some of it combines with water to form carbonic acid. But remember, carbonic acid is unstable, so that dissociates into bicarbonate ions and hydrogen ions. So going along with the high pressure of CO2, we have a high concentration of hydrogen ions. And how does that affect the pH of our blood? It makes it more acidic, so a low pH. So all of these things that I'm listing right now are triggers that can cause our normal curve to shift to the right and unload oxygen. What else? If you've been working your muscle, what's the temperature of it? It's going to be hot. And then there's one more that we haven't talked about yet. When red blood cells metabolize glucose, they release a chemical called BPG. So high amounts of BPG, basically just a signal that our red blood cells are working. So if any of these conditions are present, it's going to shift the curve to the right. And then let's take a look over here at our other dotted line. And if we compare this to this, we have a higher hemoglobin saturation. So that means there's going to be an increased affinity between hemoglobin and oxygen. And this is considered to be a left shift. That means that less oxygen will be delivered to the tissues because it's staying bound to the hemoglobin. And literally we can just go through and we can list all the opposites. So a situation with high amount of oxygen, a situation with low carbon dioxide because the tissue hasn't been working. That would also mean low hydrogen ions and high pH, low temperature, and low BPG. Hemoglobin is really good at its job of binding oxygen, because if we think about this, the tissues that need oxygen are the ones that get it. If you haven't been working and you don't need oxygen, it doesn't release. It's really like a perfect delivery system. One other thing that I want to mention is how carbon dioxide influences hemoglobin's ability to bind to oxygen. So there's something called the Bohr Effect that states that as carbon dioxide enters the bloodstream, it causes more oxygen to dissociate from hemoglobin. So if I'm trying to think about what that means, I could have a red blood cell, and let's say that it's bonded to some oxygen right there. But if we have a high concentration of CO2 in the bloodstream, that hemoglobin is going to release its oxygen. And again, that's a good thing because an area that has high CO2 has most likely been working hard and actually needs oxygen. And then the Haldane effect. The dissociation of oxygen allows more CO2 to combine with hemoglobin. They don't bind to the same spot, but the binding does cause a shape change. So once the oxygen leaves the CO2, once the oxygen leaves the hemoglobin, that allows the CO2 to bind to the hemoglobin. Pretty cool.